Hello and welcome. This video examines the details that occur during stage 2 and 3 of cellular respiration. Let's begin. Hi everyone. Today we're going to kick off where we left off from yesterday, which was glycolysis, and we're going to take those two pyruvate that was the product of glycolysis and talk about what happens with it through the next two stages of cellular respiration. So we'll be talking about pyruvate oxidation, okay, how that pyruvate transforms through oxidation as it starts to enter the mitochondria through that process. And then we will look at the glorious Krebs cycle and all of its wonderful details, all within the mitochondria as well. But first, let's talk about the mitochondria a little bit more. So we know that it's called the powerhouse. Okay, there's a nice little simplified diagram there. There are multiple mitochondria in a cell, depending on the specific function of the cell. For example, a specialized cell that is a muscle cell that makes up the muscle tissues in our body. There might be more mitochondria if that specific cell requires lots of energy to move things, for example, or maybe not as much, although the cell still needs ATP in itself. So here is a generalized diagram. In the phytosol or cytoplasm, those two words are interchangeable, for each glucose molecule consumed, only 2 ATP are produced. Okay, we saw that this is what happened in glycolysis, right? So where are the remaining 34 coming from? Because cellular respiration produces 36 ATP as the grand total. So that means those 34 are going to be made in the mitochondria during stage 2, 3, and 4, the remaining stages of cellular respiration. So how do we get in there into that mitochondria and what happens inside? So here's a scanning electron micrograph on the right side depicting the mitochondria and all of its lovely folded cristae on the inside. If you remember from our organelle presentation or lesson, there's an outer membrane that surrounds the mitochondria as a whole. And then there's the inner membrane, which creates those folds, which is the cristae. There's also an intermembrane space between the outer and those inner folds of the membrane. Okay, and within the inner membrane, within those folds, is a matrix, which again we know has like ribosomes, it has mitochondrial DNA, that is all contained within. So again, the outer membrane that encapsulates the entire mitochondria contains transport proteins, specifically something called porin, which affects its permeability, which allows for materials to enter and or leave. The inner membrane contains phospholipid cardiolipin that makes membrane impermeable to ions. Okay, and this is a condition which is required for ATP to be produced because there has to be a gradient that is produced between on both sides of that membrane. So it can't be permeable. It needs to keep them either in or out for the production of ATP, which we'll be looking at tomorrow specifically when we talk about electron transport chain and chemiosmosis. That intermembrane space, that is kind of the space between the outermost capsule of the mitochondria, that outer membrane, and the inner folds. This is the fluid-filled area that has enzymes and hydrogen ions. Then that matrix within the cristae is actually the location of the Krebs cycle. And the cristae are the folds of that inner membrane, specifically where the electron transport chain enzymes are found for that final stage 4 of cellular respiration. So let's talk about stage 2 of cellular respiration. Now, this stage actually has many different names. In our textbook, they call it pyruvate oxidation, so I'm going to stick with that. However, oftentimes this is called a transition reaction. There's only one main purpose, and it's to transform the pyruvate, okay, through pyruvate oxidation. So that's why oftentimes we'll call it pyruvate oxidation. Sometimes they'll call it oxidative decarboxylation because the pyruvate cleaves the CO2 off of the molecule, and that's called decarboxylation. But we'll stick with pyruvate oxidation. So here is a picture of what happens to the pyruvate. Remember, pyruvate is the product of glycolysis. That is the glucose molecule that has eventually split or cleaved into two, right? So remember, this picture is showing one pyruvate, but there's actually two pyruvates that are undergoing pyruvate oxidation at the same time. So we can see that the pyruvate is originally located in the cytoplasm because it is the product of glycolysis. However, this is how it's going to move through into the mitochondria through this transition reaction. So you can see that there is a transport protein embedded in the outer membrane of the mitochondria. And this kind of accepts the pyruvate and allows for it to pass through. And what happens in stage A, or part A, of stage 2 of pyruvate oxidation, is once it enters the mitochondria, decarboxylation happens. So the pyruvate breaks off CO2 from it, okay? And now it produces a 2C molecule. And remember, pyruvate is this 3C molecule, 3 carbons, because it originated from that 6C compounds, the glucose, which broke into 2. Okay, so now it's losing another carbon. It's being decarboxylated along with the 2 oxygens there, and it's formed into the 2C molecule. Then oxidation happens. Okay, so in B there on the diagram, you can see that NAD plus comes in and it forms an acetate molecule because through redox reactions, that molecule is losing an ion to NAD, making it NADH. And then part C, there's an attachment of coenzyme A to that acetate molecule, which forms a larger compound in itself called acetyl-CoA. And you could see that as the product of pyruvate oxidation. And now we're right in that matrix there of the mitochondria. So in a nutshell, in pyruvate oxidation, for each molecule of pyruvate, remember, this is showing what's happening with one, but there's actually 2 that are undergoing this transformation reaction. In this pyruvate oxidation, there is 1 carbon dioxide molecule that is released. You could see that in part A. It's highlighted in the diagram in orange. And also, when NAV picks up an electron, NADH is produced, which we know is a high energy molecule, which is also awesome. So with 1 pyruvate, you get 1 CO2, 1 NADH that's produced, and you multiply that by 2 because there are 2 pyruvates that are doing this, okay? So again, here's the grand overview of what we just looked at, the pyruvate oxidation. This is happening in the outer membrane of the mitochondria, and it transforms pyruvate into acetyl-CoA. And you can see that in the top part of the diagram there. And where that acetyl-CoA goes to is into the Krebs cycle. Okay, it is the precursor to start the Krebs cycle. And as you can see, the Krebs cycle is a gloriously detailed cycle. All right, there's a lot going on in stage 3, the Krebs cycle, and this is happening in the matrix of the mitochondria within the cristae there. What we see happening, there are, first of all, 8 steps. And sometimes the Krebs cycle can also be called, there's many different names, just like pyruvate oxidation has many names. Many textbooks use Krebs cycle, but sometimes they'll call it the citric acid cycle. It's the same thing because it starts with citrate there. So let's go through the steps 1 all the way to 8, and let's see how this is all broken down. After this lesson, I will have some videos that will explain it either through music or through breaking it down on how to memorize this, okay? I'm just going to throw this all there at you at once right now. Let's try to follow along with the steps in the diagram, and then we'll try to break it down a bit more for you. So part 1 there, in the top right corner at 1 o'clock, we see that acetyl-CoA breaks into coenzyme A. Okay, you can see the Co-A coming off. That's shorthand for coenzyme A, which is recycled. It could be reused. And an acetyl group, which is that 2C compound that was originally made in pyruvate oxidation in stage 2. And this acetyl group, the 2C compound, joins to an oxaloacetate, which is a 4C compound. And together, this creates citrate, which is a 6C compound. So there you can see the 6C compound eventually formed at the end of stage 1, that citrate. Okay, now step 2, citrate, which is 6C, converts into isocitrate, which is also 6C, so kind of an isomer of sorts. It isomerizes and converts and changes shape, but keeps its chemical formula. Stage 3, we go into isocitrate, which is we use the isocitrate, which is the product of step 2. And that isocitrate loses a CO2 and is then oxidized by NAD. We see the NAD coming in here. It's taking an electron from it. And the end result in stage 3 is alpha-ketoglutarate, which is a 5C compound, because remember, it loses a CO2 from the 6C, so that's when it reduces to a 5C compound. This is another example of oxidative decarboxylation, just like how we saw in the pyruvate oxidation in stage 2. All right, you guys still with me? Let's move on to step 4 in the Krebs cycle. So we're almost halfway there. So that alpha-ketoglutarate, which is the 5C compound, is converted to succinyl-CoA, which is a 4C compound. Okay, and this happens in 3 steps in this stage, part 4. So it's going to lose a carbon dioxide. We can see that, again, NAD is coming in. It's being oxidized. It's gaining an electron there again. And we also see the attachment of coenzyme A. Remember, we lost it off of acetyl-CoA at the very top there. It gets recycled and reused, and we see it coming back in here in step 4. Step 5. So we have the succinyl-CoA, which is the product of step 4, and it's converted to succinate. And this happens in the following way. So coenzyme A that had just joined is going to break off, and again, it's going to be recycled. A phosphate attaches temporarily to succinate and is then transferred to GDP, forming GTP, right? Diphosphate to triphosphate, because it gains a phosphate group. That GTP transfers phosphate to ATP. Okay, it takes that phosphate group that it just gained, transfers it to ADP, and now we have an ATP molecule. Bam, some power coming from that Krebs cycle. So this is an example of substrate level phosphorylation that we spoke about before. So step 5 is very important in generating ATP, although in steps 3 and 4, we also get NADH produced, which is a high energy molecule as well. We will be using all of the NADHs and the FADH2s that get produced later on in the electron transport chain, which is the next stage after the Krebs cycle. Okay, so ATP is produced. Finally, the first and only time ATP is produced in the Krebs cycle. Let's move on to stage 6. So we have succinate, which is a 4C molecule. It's oxidized by FAD to form a compound called fumarate. Okay, so you can see that FAD comes in, takes an electron, and that forms FADH2, another high energy molecule, which will be used later to eventually generate more ATP. In stage 7, you can see that water comes in and is added to that product of stage 6, the fumarate, and it forms a compound called malate. And then stage 8, we see that malate, which is a 4-carbon compound, is oxidized by NAD. We see NAD coming in again, an ion. It steals an ion from malate, and it forms oxaloacetate. That is the right word, which is regenerated to begin that cycle once again. So in step 8, it creates a product that can then re-enter the cycle once again. And we also see that another NADH, that high-powered molecule, is also generated. Phew! There it is, all at once. So let's break this down as to what the final products are. We looked at all the details. We looked at all of the compounds that they transform into, but the takeaway is what it's produced. So we end up seeing 2 carbon dioxides that are released, okay? We also see 3 NADHs produced, and then 1 FADH2. Okay, you can see those boxed in on the diagram on the left. So remember, this is showing what's happening with 1 pyruvate molecule from glycolysis, but we know that there are 2 pyruvates produced at the end of glycolysis. So this cycle is happening at the same time with the other pyruvates. So we have to multiply these products by 2. So we end up with 4 carbon dioxide, 6 NADHs, and 2 FADH2 as a grand total from the 2 pyruvates. Let's talk about carbon. Okay, where is that carbon going to? So we look at the glucose. Remember, matter can't be created nor destroyed. It's always just converted through chemical reactions. And we can see that cellular respiration is multiple chain reactions of chemical reactions, right? So we're going to see multiple changes happening with the carbon that originated in the glucose. As you can see in all the steps that we're looking at, every new molecule that's produced, we make particular points of looking at how many carbons are in that new molecule and where is it going to? Is some carbon being released as CO2, for example? So tracking the carbon so far that we've been looking at through stage 1, glycolysis, 2, pyruvate oxidation, and 3, the Krebs cycle, what we see happening is that 6-carbon glucose is then splitting in half and producing 2 pyruvates, which each have 3C. And then from those 2 pyruvates, it's producing 6 CO2s, and CO2 has 1 carbon each. Okay, multiplied by 6, that gives you 6 total, which is still the same amounts that there was in the very beginning of cellular respiration in that 1 glucose molecule that has 6 carbon. So yes, that 6 comes from the 4 from the Krebs cycle, as well as 2 total from the pyruvate oxidation. So that's the lumped sum as a total. This last slide here is going to be looking at high energy molecules produced. Okay, the previous slide was looking at carbon and what was happening to it. Let's have a look at the high energy molecules. So we often place emphasis on ATP because this is the chemical energy that directly can fuel cells. This is what cells can directly use to be able to make things happen, make work happen within the cell. So we often place emphasis on ATP. We can see that 2 are produced in glycolysis. We have 2 that are produced in the Krebs cycle. Remember, we're talking about from each pyruvate. Each pyruvate creates 1, right? Each pyruvate goes through the Krebs cycle producing 1 ATP at the end. So when we're talking about both pyruvates undergoing the Krebs cycle, that produces the 2 ATP. So all together between glycolysis and the Krebs cycle, we end up generating 4 ATP. We know that pyruvate oxidation does not generate anything, which is why it's more so seen as the transition reaction, just to transition the pyruvate into a form that can be used. But there are also high energy molecules like NADH and FADH2 that aren't necessarily used directly by cells for energy, but in the end, which we'll see in our next lesson, is used to create more ATP. So it's a precursor to create more ATP. So they're still very valuable to note as to how much are made. So we know that glycolysis, 2 NADHs are produced in the cytosol, in that cytoplasm. During pyruvate oxidation, there are 2 NADHs that are also produced. Remember, for each pyruvate, 1 is produced. So we're looking at the grand total there. The Krebs cycle, again, this is the grand total between the 2 pyruvates that enter, or I guess the acetyl-CoA that enters the Krebs cycle. So we end up with 6 NADHs and 2 FADH2s for a grand total of 10 NADH and 2 FADH2. We're going to keep this tally going as we look at our next lesson tomorrow at the electron transport chain. How many more ATPs will be produced? We already know, we kind of just talked about that in the beginning of this lesson, but how does that all happen? We'll get into that next.